أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم ويقولون متى هذا الوعد إن كنتم صادقين ما ينظرون إلا صيحة واحدة وتأخذهم وهم يخصمون فلا يستطيعون توصية ولا إلى أهلهم يرجعون ونفخ في الصور فإذا هم من الأجداث إلى ربهم ينسلون صدق الله العلي العظيم. Function of nephron for second year class in 12th lecture. Nephron, structure of the nephron, I already started in previous discussion. What is nephron? It is the cell situated in the kidney. Nephron is doing very tricky works because it is absorbing the urea from the blood, digested food products, sodium ions, useful salts, but by tricks it brings back to blood the useful things and remove the nitrogenous waste component, which is the urea. We are ureotelic living thing. I am not telling animals, but animals are written in the book, but I'm not telling human being, animals in this case. I will say ureotelic human being. They are the ureotelic. We go back to the structure of nephron. I just want to recall the structure of nephron, because structure of nephron I am going to tell you once again what is happening. The two part of nephron, I will name the two part of the nephron. One is the renal tubule. This is the first part of the nephron. Renal tubule. This is the first part of the nephron. Second part of the nephron, I will say renal corpuscle. I will show you slide. RENAL CARPUSCLE, renal corpuscle. This is the renal corpuscle equal to Bowman's capsule. I am dividing the nephron into two major parts. You will feel easier when you will divide the nephron into two major parts. It is easier to study. Renal corpuscle. Where is the renal corpuscle? I am going to tell you. These are the spellings. I already made the spelling renal corpuscle. Second one is the renal tubule. So two words I am telling you, renal corpuscle and renal tubule. Two parts of the renal corpuscle, renal tubule, two parts of the nephron. So first part of the nephron, which is the renal corpuscle, equal to Bowman's capsule and the glomerulus. What is the glomerulus? You know, I was talking in the last session that efferent arteriole, renal artery goes to efferent arteriole goes to network of the capillaries. These capillaries are more modified and they don't go to venules. They have large pores and they have pressure filtration. By pressure filtration they can bear more pressure, and by pressure they send the useful and the harmful things into the Bowman's capsule. This is the important action in the nephron, and nephron main function reside with Bowman's capsule specification and glomerulus modification capillaries, and they don't go into venules, it go into efferent arterioles, which I will tell you in the diagram. So Bowman's capsule and the Bowman's capsule, glomerulus, network of the capillaries with specialized pores, with specialized structures. In A levels, the Bowman's capsule and the glomerulus are so much in detail that I need to explain for 1 week, and because these informations will tell how the food, digested food and urea by pressure enters into Bowman's capsule. So these are then become the renal tubule. The second part, the renal tubule, and renal tubule, first part, proximal part. This is the word which I will explain. The first part of the renal tubule will be the proximal convoluted part, the twisted part. Second part, descending Henle's loop, and then ascending Henle's loop. Henle's loop will come under one heading, and then third part, that is the distal part. Distal part is the last part of the nephron, and many nephrons open into a major duct, collecting duct, and collecting duct enter into another little larger duct, papillary duct, and papillary duct opens into renal pelvis. I was telling you that nephron is not the structural unit of the kidney. Reason is that the cavity of the kidney don't consist of nephron. Only two parts of the nephron consist of the kidney. In the last lecture I was telling you, cortex and the medulla. These are the two structures which consist. consist of nephron. Only two parts of the kidney consist of nephron, cortex and the medulla, not the renal pelvis. So collecting duct actually enters into a cavity in which all urea and the urine, where is the useful products, they went back. How they went back, this is the discussion we have to do in this chapter and in this lecture. So I am interested to tell you a significant capillaries, the peritubular capillaries. If I am telling you that reabsorption of digested food take place and the food which goes into Bowman's capsule by pressure filtration, I am showing you diagram, and by diagram you will be able to understand in a better way. And if I am telling you that the useful components like amino acid, glucose and the fatty acids and the cholesterol, even the nitrogenous waste component, the urea, they enters into Bowman's capsule by pressure filtration method. The useful components will be absorbed. The credit goes to proximal part of renal tubule. Number one, I will prefer proximal part first of all, but second credit I give to peritubular capillary. The peritubular capillary, these are the capillaries. So peritubular capillaries, they actually absorb the useful components once again, and they are formed by division of efferent arteriole. You are getting the reason also why glomerulus capillaries don't become venules according to rules of circulatory system. They must change into venules. They must enter into venules, but they did not enter into venules. I am talking about glomerulus. They enters into efferent arterioles. The reason was that because they just wanted to break once again into the capillaries, and because digested food has been lost, so digested food has to be absorbed by the peritubular capillaries from the area of the proximal part, first part of the renal tubule. Endless loop are doing very specific functions, and functions of the kidney are more important, because when you will see this one, you will be able to understand everything which I just want to tell you now. Look here, I want your attention, because every function, each function you will be able to understand here. Look here, this is the efferent arteriole. The blood is coming. This is the renal artery. Blood is coming here, blood is here. It will go to network of the capillaries, the glomerulus, the network of the capillaries. It is surrounded by a capsule, which is the Bowman's capsule. I was telling you this area is so important, that this network of capillaries, glomerulus, and this area which is a Bowman capsule, it is so important that it is modified with different names, with the different structures, with the different mechanisms. I am already telling you that in A levels this information is much in detail, and I will provide you further details when syllabus will finish. Extra information I will provide. You will be happy to know further about the layers of Bowman's capsule and the layers of the glomerulus, and how they are able to send useful component from here to there, mean from here to there. It mean from glomerulus to Bowman's capsule, glomerulus to the Bowman's capsule, the digested food also enters, nitrogenous waste component also enters, and sodium salts also enter, some other useful things also enter. Therefore, now this Bowman's capsule, which process? The pressure filtration process. How the pressure filtration process take place? The modification of the glomerulus, the modification of the Bowman's capsule, I am already told you, because this modification due to different membranes, different structures, microscopic structures, because these are not the normal capillaries, these are the modified capillaries. Bowman's capsule is modified, and magnifying structure, which I will tell you if you wish, but when the syllabus will finish. Okay, come back. Efferent arteriole, this is the efferent arteriole, and this glomerulus by pressure enters the useful component, once again I am telling you, into the Bowman capsule. Now this tube, which is first part of renal tubule, this area I was telling you, renal corpuscle, glomerulus plus Bowman's capsule, they both will called as collectively renal corpuscle. Now go back to the structure which I just want to tell you, renal tubule. This is a renal tubule. This is the first part, proximal part. This is yellow color, descending endless loop, descending endless loop. This is ascending endless loop. This is distal part, Once again, okay, just hold on. I just move to figure to magnify to show you exactly the renal tubule, the first part of the renal tubule, the descending handless lobe, ascending handless lobe, distal part, collecting duct. And collecting duct is not the one duct for nephron. No, many ducts are opening. Look at the area of collecting duct in which the nephron will open, another nephron will open. So nephron, what the nephron is doing? Problem with the nephron here at this location. In this location, nephron has useful components as well as the harmful component. The nephron has the issue. What is the issue? Nephron need to absorb the digested food from here into the peritubular capillaries. I will tell you where are the peritubular capillaries. Yes, of course. Efferent arteriole. This is efferent arteriole. After glomerulus, efferent arteriole. Why there is need of efferent arteriole? Reason to absorb the digested food and the useful component once again into the blood. It break down into the capillaries, red color capillaries, the peritubular capillaries. Useful component now goes into peritubular capillaries. Similarly, some useful digested food and some water is also absorbed, and here descending handless lobe, the water is absorbed into the capillaries which is surrounding the handless lobe. The capillaries surrounding the handless lobe is called vasa recta. This is the vasa recta. This is not the peritubular capillary. When it is surrounding distal part, when it is surrounding the proximal part, when it is surrounding the distal part, such capillaries are peritubular capillaries. And when they are surrounding, I have to finish this one and then I have to indicate you for again. When they will surround the handless lobe, ascending handless lobe, the descending handless lobe, it means that is that capillary which is surrounding the handless lobe, that is called the vasa recta, V-A-S-A-R-E-C-T-A, vasa recta. Vasa recta, what is the vasa recta is doing? It is absorbing the water from descending handless lobe, mean it is going from the tube to the blood capillaries, you mean vasa recta. How it is going? It is going by counter current multiply. That I think is not included, but I will tell you what is in simple fashion. I will tell you what is the counter current multiplier mechanism. That the water is moving in lower direction in the yellow tube, mean descending endless loop, and the blood capillary, mean vasa recta, is moving the blood in the upper direction. In opposite direction they move in. When they will move, both liquids are moving in opposite direction, the water will come out from descending endless loop to the vasa recta. The counter current multiplier, the both flows are opposite, so that their movement of the water molecules from the descending endless lobe to the vasa recta becomes little easier. Now I am telling you two important hormones which are involved in the nephron. Look at the brain is controlling this, because this is homeostasis. Nephron need a boss. The boss is controlling the nephron. How it will be controlled? The two boss is the hypothalamus, and boss is the P gland, the pituitary gland. It is secreting two endocrinal hormones. One endocrinal hormones will come here in this location. That is, what is the location of this part? You know, this ascending endless lobe. Of course, ascending endless lobe. What happens to ascending endless lobe? The sodium ion will move from ascending endless lobe to the blood capillaries, mean peritubular capillaries or the vasa recta. You are feeling confusion in the peritubular capillaries. I know you are feeling confusion and you wanted to know, sir, what is the difference between peritubular capillary and the vasa recta. I will tell you nothing difference. You will say vasa recta as a peritubular capillary. Reason I tell you, this all area surrounded by peritubular capillaries. This all area is a peritubular capillary. This all area is the peritubular capillary. Just the peritubular capillary, when they come under the handless loop heading, under the handless loop, they become the vasa recta. They become the vasa recta. So there is no much difference. There's no need to confuse. You can say even he is telling peritubular capillary. Vasa recta, I am just telling you that vasa recta and vasa recta at the handless loop, it is called vasa recta, but it is branches are still called peritubular capillaries. So there is no major difference between the peritubular capillaries and the vasa recta. Go back to third part Aldosterone I told you, yes. Aldosterone absorbs the sodium ion from the ascending NS loop. Now I have to move the third part. It is little twisted. Look here, two parts are twisted in the nephron. The proximal part is a little twisted and the distal part is a little twisted. The twist is just because of the reason that they will absorption power should be more, because food is being lost, the water is being lost, the salts are being lost, and tubular secretion mechanism I have to tell you in the next session. That is also a significant part of the nephron. Here I will tell you once again the water will be absorbed, but not by end collecting duct, but not by counter current multiply, by hormone which is coming from the brain, by the blood. Endocrine hormones move by the blood, and those hormones are called endocrine hormones. The name of the endocrine hormones which absorb the water from the distal part and the collecting duct, this is the antidiuretic hormone. Anti mean against, I mean removal, uretic mean urine. It is not urine being removed, water is being removed from the tube distal part to the peritubular capillaries. So it means that antidiuretic hormone is maintaining the water concentration. It means that nephrons are not removing urea only, they are maintaining the water concentration. It means that the kidneys are not only excretory organs, they are also osmoregulatory organs. It means that nephron is not removing urea and maintaining the water concentration by ADH, it is also maintaining the salt concentration by aldosterone. It means that nephron is not removing urea, not absorbing sodium, not absorbing water only, it is also absorbing useful components, digested food once again into blood. So you can understand the tricky role of the nephron. That was the point I was talking you in the last lecture. Collecting duct opens into papillary duct into renal pelvis. What is the location of nephron? This nephron present where? In the which part of the kidney? Cortex and the medulla. So 2-3 slides I just want to show you of today's lecture, and one slide I would like to show you. Nephron is very important part, integral part, and I will tell you the 2-3 things more about the nephron. I was talking efferent arteriole. Yes, efferent arteriole. This point, students, you have to listen very carefully because this is little technical, little more technical. Look here, you will understand. I am just magnifying the efferent arteriole. Magnifying efferent arteriole. Okay, ji. This efferent arteriole, you are watching the lumen of the efferent arteriole. What is the lumen? Cavity. The cavity is more. Just confirm me. Just shake your head, yaar. Just shake your head. Okay. The cavity of the efferent arteriole is more. Clear? Go back now. The cavity, this is efferent arteriole. The efferent arteriole and cavity of efferent arteriole, just tell me. It is more or less. Cavity of efferent arteriole, just tell me, it is more or less. It is less. Of course it is less. Of course it is less. So efferent arteriole cavity is less. Efferent arteriole cavity is more. I am telling you pinpoint why pressure is more in the glomerulus. When here the volume is less, lumen is less, here the lumen is more, here the blood flow can easily, here the blood flow by difficult way. When it goes to glomerulus, next lumen is less. Before glomerulus the lumen was more. This is glomerulus, here the lumen cavity was more, here the cavity was less. What happens? The pressure increase here. The pressure increase here. When the pressure increase here, what happens? Pressure increase, this pressure push the blood by force, the useful components, the urea, red blood cells, white blood cells, protein will not go, fibrinogen will not go, prothrombin protein, clotting protein will not go, because nature don't want to disrupt the body, damage the body. Only the things which will go, which can come back into the blood. The digested food will go, the urea will go, some salts will go, and water will go, which can be reabsorbed. And urea, which is our enemy, will be able to remove from the body by help of this collecting duct. Thank you very much. And this was lecture, and I will come very soon with the lecture number 13, but I will continue discussion about excretory system. Allah.